My name's Megan. I'm the research coordinator for GRICon, and I'll be moderating this session. So today we are joined by Dr. Lonnie Wollmuth, who is in the Department of Neurobiology and Behavior at Stony Brook, and this work is in collaboration with Dr. Howard Sirotkin. He's going to talk to us about zebrafish studying brain development and NMDA receptors. So we're really excited to hear from Dr. Wollmuth. And if anybody has any questions, you can log in the app and ask your questions there, or we'll also have time for questions at the end.
So thank you, Megan. Yeah, I'm going to talk to you today about zebrafish. And I always like to start off with this slide first, just to emphasize my background's in NMDA receptors. So I'm not a zebrafish geneticist, but I've become a zebrafish geneticist. And that's mainly because of this individual, and also partly due to this individual right here. So we have several people working on our zebrafish project, but Josiah Zuidsman was sort of the pioneer and one of these outstanding graduate students who kind of help you get a project going. And he's now moved on to a postdoc at UPenn. But again, he's a zebrafish geneticist, although I now call myself a zebrafish PI. So the reason we're at this meeting is because we know now that there's a lot of variants, mutations, either missense or nonsense mutations in genes encoding NMDA receptors. And these mutations are associated with a variety of clinical outcomes, right? Epilepsy, intellectual disability, autism, muscle or movement disorders, and other sorts of disorders like schizophrenia. And when these are first identified in the patients, I think there's sort of four general questions we always want to ask. And the first one is, does the variant contribute to the clinical phenotype? And this entails really just sort of basic characterization of whether, especially for missense mutations, whether that change in the amino acid side chain changes the function of it. Is it a loss or gain of function? There's also questions sort of just from a structural function perspective is, what's the structural basis for how the variant causes alterations in receptor function? But really what I'm going to talk to you today are these two topics here, mainly this one here. And then the question is, how does the altered function of the NMDA receptor contribute to the clinical phenotype, one? And two, how to treat clinically the individual or family with the variant. How to really, after meeting families and seeing the patients, really, I think the first step that would be beneficial to them is improving the quality of life. And that in many aspects of that. But when we think about glutamate synaptic transmission, as I've done throughout my career, I've been working on this for 25 plus years, I think about glutamate in the sense of being present at a synapse, right? And then we get this presynaptic action potential that causes the vesicular release of glutamate, which is represented in black, that interacts with these postsynaptic receptors, AMPA and NMDA receptors. And then that leads to this biological signal. Typically, we think about it in terms of ionotropic function. And what I think I want to emphasize today, and I think we've seen it in some of the earlier talks today too, is we've got to go beyond this role of NMDA receptors as a synaptic protein, that it's going to be involved in many aspects. And the one aspect I'm going to emphasize today is the role of NMDA receptors in brain development. Sorry, my thing didn't hit. So again, we're going to focus on these two general questions today, and we're going to use zebrafish as a model. And zebrafish offer many advantages, and I'm not going to go through all of them, but I just want to emphasize three of them. One is rapid development, right? So they undergo very rapid development, and again, I said we want to understand the role of NMDA receptors and disease-associated variants, how they're involved in brain development. So this is a real advantage. They also display really well-characterized larval behavior. And we can use this larval behavior as at least an initial representation of brain circuits and dysfunctions in those brain circuits. So it's a very straightforward way to do that. And then we can delve into deeper as to why this might be changed. And the last one is these high throughput drug screens. And I'm just going to touch on this because this is on our agenda, but we're really not there yet. And so what's the advantage of zebrafish in this regard? Well, first of all, zebrafish absorb drugs across their gills, right? Second of all, depending on the behavioral assay, you can do up to 96 animals at once. So the potential for high throughput screens, it far exceeds that with what you can do with a rodent model, for example. Again, we're not going to get into this today, but it's really high on our agenda to start moving forward to do that. And again, so what I'm going to tell you is, okay, so we're interested in zebrafish. We're using zebrafish to understand NMDA receptors and disease-associated variants. Ultimately, we want to start doing high throughput drug screens using sort of 96 well plates where we can sample a lot of drugs. But what I kind of want to talk to you today is a sort of a different aspect. What we're really also using zebrafish is to sort of understand how alterations in NMDA receptor signaling lead to disrupted brain circuits. And we believe, as well as many other labs believe, that understanding how these disrupted circuits arise may lead to novel therapies. Again, maybe we need to think beyond NMDA receptors, PAMs and NAMs and pore blockers and start thinking about other systems that get disrupted in this process. So just to give everybody a little bit of guidelines for zebrafish. So again, you're gonna hear me reference periodically this days post-fertilization. So that occurs at day 0. And again, this is an egg. So all this process occurs externally. So it's much easier to study than in rodents. And you have this 3-day period. It's called the embryonic development period. And the egg goes from this fertilized egg through this whole process here. And at the end of day 3, you're at this state here. And then at day 3 forward, the zebrafish display a lot of well-defined behaviors that can be characterized. And the one I'm going to talk about today is social interaction. So again, social interaction deficits, they're hallmarks of neurodevelopmental disorders. So we're going to use that as a hallmark of disruptions in the signaling in a brain circuit as a first step. So one of the disadvantages of zebrafish, about 3 million years ago, they had a gene duplication. So rather than having just one gene, as we have in humans, you actually have 2 paralogs. Now, and that occurs for these major subtypes of GRIN. Now, in ways that can also be advantageous. So in rodents, knockout of GRIN1 is embryonic lethal. So you can't study it. It turns out if you knock out GRIN1A, they're viable. GRIN1B, these 2 paralogs for GRIN1, they're also viable. And if we cross these 2, in which case we have no NMDA receptors in the animal, they survive till 10 days post-fertilization. So it gives us an opportunity to study the general role of NMDA receptors in brain development. And we published on that. We have 2 more manuscripts coming along, working on that. But what I'm going to talk to you today about is our work with GRIN2B here, okay? And tell you this story here that we just published this year. But before I get there, I want to give a little more background on this. And the question is, are zebrafish a good model to study human variants? So first of all, what we've done is we've done a sequence alignment. So again, this is in terms of those familiar with the NMDA receptor structure, you have these 4 layers. So this is just one of the subunits extracted out. You can see the amino terminal domain, the ligand binding domain, the transmembrane domain, and this intracellular C-terminal domain. So in terms, we did sequence alignments between the human subunits and the zebrafish paralog. So from about here, all the way to the end of M4, everything in terms of sequence similarity is close to 100%. So there's a really high sequence similarity. In the amino terminal domain, there's a little less sequence similarity, but again, the key functional domains in the ATD are highly conserved. Now the CTD gets a little more, less conservation, but it turns out, I just want to point out 2 features of this. The first is that in terms of non-rodent models, or in humans, the GLUN2A CTD is huge. It's very large. It's humans way back here. And zebrafish also have this very large CTD, so just like the humans. And what's important here, and I'm not going to show it to you, is that when you look at the key structure function motifs in the C terminus of these two subunits in particular, they're highly conserved. Domains like the CAM kinase site, the last 15 amino acid residue, palmitylation sites are conserved. So the key structure function motifs seem to be conserved. And the last thing we did is we went to GRIN portal and we identified unique missense mutations, okay? And we've identified, these again are just unique missense mutations. So there's 64 in GRIN1, 43 in GRIN2A, and 67 in GRIN2B. And you can see these missense mutations are extremely highly conserved in the zebrafish paralogs. And in fact, the only divergence occurs either in the signal peptide or the extreme amino terminal domain. So given this bits of this evidence, and I'm going to show you a little more in that, we're going to argue that zebrafish NMDA receptors are a good model to study human NMDA receptors. Okay, so now I'm going to move over to the story about GRIN2B. It's well known. If you go to Safari, that website, they'll talk about GRIN2B as being a high-risk gene in autism. So we went in, and again, here's another way we verify the similarities between zebrafish and human NMDA receptors as we characterize them functionally. So we've taken the zebrafish GRIN2BB and the GRIN2BA. We're not showing it here. And one of the hallmarks of GLUN2B is it's got this really slow deactivation in response to brief applications of glutamate. And you see, this is what you see in the human subunits, and this property is conserved in the zebrafish. So again, you see a lot of not only structure conservation, but also functional conservation between this. So we used CRISPR-Cas. Again, we can inject the reagents into these cells. We put in lesions in the M3 transmembrane segment. And then what happens is you generate these early stop codons. And then we do a bunch of these, and then we decide to take the one that's the earliest stop codon. And when we look at antibodies, there's no GLUN2B detectable in these brains. Now, one of the differences is, again, in rodents, so I should say we did that for GLUN2BA and GLUN2BB. We cross those animals. And these are what we call the GRIN2B minus. So these are, again, animals without any GLUN2B subunits. Now, in the rodent, they die, and it's believed that they die because of a suckling deficit. So again, here's an advantage of zebrafish. They don't have to suckle, so presumably they're able to live to adulthood, and we can cross them and carry on experiments with them. So here's this social preference assay that we're using. And again, it's got 10 lanes in it. We can just focus on one of these lanes here. And so this fish here cannot see the fish in there. So really, it's just what occurs in the lane. And at the end of each lane is a glass here and here. Here it's empty. And here we have a con-specific animal here. Okay, so we put fish in there, and then we randomly or not arbitrarily divide this into 4 zones. So there's zone 1, 2, 3, and 4, with 4 closest to the con-specific. So you can see at 1 week post-fertilization, or 7 days post-fertilization, the fish pretty much spends its time randomly in these 4 lanes, or these 4 zones. By 2 weeks, they're still showing some pretty random, they're showing a preference here. And then by 3 weeks post-fertilization, they're showing a significant preference to occupy the lane adjacent here, and this effect gets even stronger. And this had been defined before, that zebrafish display this strong social preference that develops and reaches sort of maturity at 21 days post-fertilization. So again, that result I showed you there is just replicated here. We're now displaying it using what we call the social preference index. So 0 means there's no preference. 1 means they're spending all their time in zone 4. And you can see with development, there's no preference here, and then that the preference starts developing in wild type. In contrast, in these GRIN2B fish, so these fish who do not have the GLUN2B subunit, they do not develop social preference, okay? And And again, this idea is, this is again just another way to represent the data and a little different. Again, you can see, so we can compare statistically. And again, we see this lack of development of social preference in these GLUN2B knockout animals. Now, we tried this same assay with another high-risk gene in autism, fMRI1. There also we see a social deficit. Notably, however, in any of the other NMDA receptor genes that we tested here, the paralogs, none of them show this social preference deficit. It's very highly selective for GRIN2B knockout. Okay, so now we've identified a behavior. And sorry, sir, I have a movie I want to run here. And so now we've identified a behavior, a deficit that says some part of the brain is no longer developed properly. And sorry, I should have pointed this out earlier. And we're going to label now in the brains, and this is again one of the advantages of zebrafish is we got transgenic lines that express a red marker in the V-glut or excitatory neurons, and a green marker in the DLX minus neurons. Are you able to get that movie going there, sir?
You should be able to forward right into it.
Okay.
Not taking for you?
Oh, I did it. I may have walked past it. No. Anyway, if we can't do it, it's no big deal. It's okay. Should I hit my button now?
It's not initiating the movie.
It's the movie?
The cue forward is not initiating the movie. Is it embedded into the presentation?
It should be. It's okay. If it doesn't show, it's okay. What you would see if you go through this movie is it would display a zebrafish brain, and it would just start from the ventral side moving down through the dorsal side, sorry, moving down through to the ventral side. And you would see a lot of red, you would see a lot of V-glut, and you would see a lot of green, right? So we see a lot of that, both red and green, both glutamatergic. But we went in and then quantified the expression using this brain browser. And this again was done in collaboration with Harold Burgess at NIH. And what we identified was that there was one brain region, the subpallium, where we saw a dramatic reduction in the number of DLX neurons. So now DLX, again, is a marker for GABAergic interneurons. And again, we can just see this. I'm not going to go through this in detail here. This we're now labeling. We're using a little different labeling or color scheme here. We're just identifying the cell-rich region in white. So again, here's the subpallium. And then here's the preoptic area. And these are going to be two critical areas. This is neural pill here, the magenta areas here. And in both of these two areas, in the subpallium and the preoptic area, we see a dramatic reduction in the DLX. So in the rest of the brain, we see no change in the number of excitatory neurons, but we see this reduction here. And this region in the brain, there's two interesting things about this region. One, in the mature animal, the subpallium it's kind of always hard to draw an analogy to human brains, but it's the part of the brain that's implicated in the social interaction, okay, in the mature animal. And two, this region is the region where it's the major birthplace for GABAergic interneurons. So this is where most of the GABAergic, and it's analogous to the lateral and medial geniculate in rodents. So somehow it appears that the presence of GLUN2B has at least one function, is to disrupt the development of GABAergic interneurons. Now, again, those who are familiar with GABAergic interneurons know there's a wide variety of different GABAergic interneurons here. And which of the particular subtypes are disrupted, we don't know. Studies are ongoing to attest that. But again, what I want to emphasize is it may or may not be the glutamatergic input to these interneurons that important, but what is clear is we're seeing a fundamental change in the population of these neurons. And that may or may not contribute to this social interaction deficit. So again, now it's suggesting maybe we should also think not only of glutamatergic signaling, but also GABAergic signaling in treating patients in particular. There's many other questions that come up. So these DLX interneurons, they're located in the right place. Whether they're directly involved in the social interaction deficit, we don't know. And it could also be that GLUN2B impacts other neuronal populations that have been shown to be involved in the social interaction, including cholinergic, dopaminergic, and oxytonergic neurons. And now we got to come to this one critical point. So what I've showed you up to now is a knockout, right? It's a complete loss of function of GLUN2B. And really, that's not what's going on in patients. And really where we want to start getting to is adding in these disease-associated variants, introducing the loss and gain of function to see whether they, in particular the GLUN2B, the loss or gain of function, to see whether they impact interneuron development and social interaction. And to do this, we've introduced into the lab what's a technique called prime editing. And when I say we introduced into the lab, that was Howard Sirotkin's. Again, he's the geneticist. And the advantage of prime editing is it combines a Cas9 nickase with a reverse transcriptase. And this allows basically, so CRISPR-Cas is very precise in its targeting, but very imprecise in its repair mechanism. And the idea behind this, and really this whole technique was developed by Keith Wong here at Harvard, was to basically fuse the nicking and the repair mechanisms into one unit. And again, we're keep trying to optimize this approach to improve it, and we're changing the PEG RNA, the templates. And we've now got, I think we have 8 of them here, 4 of which are now apparently in a germline. So we've now got 4 of these human disease-associated mutations. This is in GLUN1, we have 3 there, and then we're now 5 in GLUN2B to study them. So we seem now to be able to have them in the germlines. And again, that's a huge step because now we can grow these up, cross them, and verify that there's no additional mutations around there, right? That's always the challenge. Yeah, we could introduce a particular mutation, but we don't want any spurious mutations. And then we want to take these fish and really ask the sort of the same questions we've already done with GLUN2B, right? We want to come back and say, how do they impact the development of GABAergic interneurons? How do they impact social behavior? Again, with the idea is if we understand fundamental processes that they disrupt, this can lead to potentially novel therapies, again, targeting specific neuron populations like GABAergic or GABAergic signaling or dopaminergic or cholinergic, et cetera. And in addition, now we have these animals, then we can start going through these high throughput drug screens that we would like to move forward with. So again, just sort of quick takeaway points. Zebrafish are a good model to study human NMDA receptors. GLUN2B regulates in an unknown way GABAergic interneuron development. Experiments are very much involved addressing that right now in our group. GLUN2B is in an unknown way is critical to the development of the social interaction circuit. We don't know if GLUN or GABAergic interneurons directly are involved in this or whether it's the development of cholinergic, dopaminergic, oxytonergic neurons. And some results I'm not going to show you here, again, is that the NMDA receptors are involved in the timing of neuron maturation. And one of the things we're discovering, as I think many people realize, is again, we think of NMDA receptors as synapses. They're everywhere in the brain and in the body, right? And we're seeing that it's involved, for example, in regulating neural crest development, which encodes or generates many non-neuronal type tissues. So again, I'd just like to thank Howard and Josiah. Most of the work I showed today was with Josiah. Amalia is doing a lot of work. She's joining my lab now as a postdoc, extremely talented, and she knows the anatomy. It's nice to have, I'm an ion channel biophysicist, and now I have to learn anatomy. It's fantastic to have someone with this talent. And then there's Erica Nebbit, who's joining the lab to start expanding on the physiology of the impact. And that's it. Thank you very much for your time.
Hello, I see we have people who are at the back there. You guys can file in and take seats. And if anybody has questions, I can repeat questions for you. talk about.
Nice talk. Development of these interneurons, but did you? Sorry, I'll start from the beginning. Nice talk, Lonnie. You're talking about the development of these GABAergic interneurons, but did you ever patch them to see what the electrophysiological properties are like?
We haven't patched them. We're setting up to do light sheet microscopy to start out imaging calcium in them, right? So to see they're basically the NMDA receptor. So one of the disadvantages of zebrafish is that it's hard to patch individual neurons. So we're actually getting a Multner cell prep going in the lab, which will allow us to actually look at synaptic transmission. But in terms of looking at GABAergic interneurons, looking in the brain, we're going to predominantly use light sheet and calcium imaging. And we have GCamp fish going for that.
Thanks.
Any other questions? Oh, I see a question over there and over here.
Oh.
Hi, thanks. I am a little bit confused about how you introduce a human mutation into the zebrafish because there is 2 different genes for the same. So if you can explain.
That's an excellent question. So that's an excellent question. So really, as we know, in humans, there's 2 genes, and one of them is the only one affected, right? What we are doing, we're going to introduce it into predominantly gluN2B, B, and then we're going to cross it with so we're going to introduce it into one of the BB subunits, and then we're going to cross them with a gluN2B knockout. So they'll only have we'll have the 2 genes in there, one of which will have the disease-associated variant, and the other one that has the wild type. And in fact, the priority experiment is to start looking at sort of a gene dosage effects of having just one subunit, initially looking with all 4, and then gradually narrowing it down so you only have what occurs in the human, okay? I think there was a question over here.
Excuse me. Some of us are intrigued by the non-neuronal NMDA receptors, in particular the ones expressed in the hematopoietic system, and we want to use potentially that expression to try and see if we can look for biomarkers. I just wondered if you had an opportunity to look for expression of green genes in zebrafish embryo and larvae to check if they express in the developing hematopoietic domains or types of non-neuronal areas.
So I hate to say it, we've looked at the expression in neuronal tissue, but now, I mean, that's a whole new project. The Neural Crest is a whole new project that someone is taking over in the group, and they'll come back to that to see expression and whether it's an autonomous or non-autonomous action, but it's clear that the NMDA receptors are having effect in non-neuronal tissue in these zebrafish.
Thank you.